Um, this is our first virtual seminar of the year, and of course, just like everything else, it's started in a little bit of a bumpy way, but we'll do the best we can. The storm is also raging here on the coast, so I'm hoping with all of my heart that we don't have any glitches in internet or power, but we'll do the best we can there as well. Hopefully you are somewhere with good food and drink. This is a Science on Tap, even if it's a little bit different this year. My name is Cinnamon Moffett. I'm the Research Program Manager at Oregon State University's Hatfield Marine Science Center in Newport, Oregon, and I will be your host for this evening. Even with all the glitches, this is a webinar format, which means that you do not have mics, cameras, or screen share abilities, but we do hope that you ask lots and lots of questions, and you would do that using the chat box that you might find at the bottom or the top of your screen. You click on the little pop-out box, you'll get a little pop-out that allows you to chat with us all. So feel free to put anything you want in there and ask any questions at any time. Wanted to let you know, as you've heard us chat a little bit, this is going to be recorded, and I'm going to, throughout this point, put in some information into the chat box. So right now, I just put in the link again. So if you needed to share that with anybody else, feel free to do it. The recording will be available on HMSC's past seminar page. So if you just Google HMSC, go to Visit Hatfield, go to the seminar page, it will be there in a few days. Couple of quick announcements. I wanted to just promo next month's Science on Tap. I'm hoping it goes a little smoother. That will be February 17th at 6 p.m. We have Charlie Plybon, Michael Moses, and Andy Lanier talking about Life on the Rocks, and talking through the process of updating the Oregon Rocky Habitat Strategy and ways that you can engage in that updating process. So excited to learn a little bit more about that. But since we had a little bit of a delay, I'm just going to jump right in and welcome tonight's speaker. We have Sara Hamilton, who is currently a PhD student and an NSF GRFP fellow at Oregon State University. She has her undergrad degree in biology and gender studies from Bowdoin College in Maine. And her current PhD work at OSU is she's taking an applied and interdisciplinary approach to studying the bull kelp forests in the Pacific Northwest, creating science that looks at many parts of the kelp forest ecosystem, including population dynamics of bull kelp here in Oregon. She is passionate about coastal management and policy work, and does a lot with Nature Conservancy and the Oregon Kelp Alliance to support kelp forest monitoring science and decision making. And Sara would like to acknowledge that she is currently living on the traditional lands of the Kalpuya people who were forcibly removed from these lands to reservation in the mid-1800s. Today, many of the descendants of those people are members of the Confederated Tribe of Grand Ronde Community of Oregon and the Confederated Tribes of the Siletz Indians. So just want to recognize that we are on those lands. And we are so excited to have Sara here and to share some of this groundbreaking work with us. And so without further ado, Sara, I'm gonna hand it off to you. I'm gonna mute myself and turn off my camera so you are highlighted, and you have the floor, and then we'll work through questions at the end.
Okie doke. Awesome. Thank you so much, Cinnamon. Thanks for your flexibility, everyone, in making it here tonight. I'm really amazed that 50 of you managed to find your way here despite the technical difficulties. So I'll try to make it worth your desperate last-minute scramble. So as Cinnamon said, hi, everyone. My name is Sara Hamilton, and I'm a PhD student at Oregon State University. And I'm here today to talk about the status and conservation of the sunflower sea star. And I want to acknowledge that I'm here today speaking on behalf of the Pycnopodia Recovery Team, which is a large group of folks that have been working together to combine data on the sunflower star to better understand how their populations are doing and to explore options for conservation. So I just want to point out that this presentation is the product of a large multinational team effort. So without further ado, let's jump in. So if everyone could humor me for just a second, close your eyes for a moment and think of the word conservation. What comes to mind? You can open your eyes, and if I had to guess, I would guess that for many of you, critters like pandas, elephants, or tigers came to mind. Now, these big, beautiful, intelligent mammals, often known as charismatic megafauna, are often what people think of when they think about conservation. So I ask, how often do you think of slimy, creepy crawly invertebrates inching along the ocean floor? Probably not very often, but I think that you should think of those when you think of conservation. Invertebrates, which when I say invertebrates, I'm talking about animals that don't have a backbone, such as insects, worms, crabs, sea stars. They actually make up more than 95% of the world's animal species. They also often play critical roles in our ecosystems and cultures and are often understudied compared to vertebrates. The sunflower star is a good example of some of these points, and I think that the case of the sunflower star has a lot to teach us about conservation. So I'll be circling back to this theme at the end of the presentation. So just to highlight what's coming here in this presentation, we're going to start with an introduction to the sunflower star. We're going to talk a little bit about the relationship between sunflower stars and kelp forests. We're going to move on to what happened to them and where are they now. And then we're going to look forward ahead to the future a bit and talk about conservation options and what does this mean for us here in Oregon, which is where I'm giving this talk from. Just as a note, I'm using the species common name, sunflower star, throughout this presentation. However, I'm really used to using its scientific name, which is Pycnopodia. That rolls off my tongue so easily these days because I use it so much. So if you hear me using that term, I'm just referring to the sunflower star. I hope that's not too confusing for anyone. So let's meet the sunflower star. The sunflower star is the world's largest sea star, growing to be up to 3.5 feet in diameter. Additionally, they're one of the fastest moving sea stars we know of. They grow to have more than 20 arms and are voracious predators. So they're basically the tigers of the ocean floor if tigers had 20 arms. So sunflower stars are generalist predators, which means they can eat a lot of things, but some of their favorite foods are clams, urchins, mussels, and snails. And you can actually see in the picture here at the bottom of the screen, this sunflower star is actually eating an urchin, and you can actually see a few purple spikes kind of popping out. Speaking of eating, one of my favorite sea star fun facts is that sea stars actually eat by pushing their stomachs out of their body, enveloping their prey in that extruded stomach, digesting them externally, and then sucking the juices up. Pretty cool, huh? So overall, the sunflower star is a pretty hardcore, badass animal, in my opinion. Now over here on the right, I have their range highlighted in purple. Historically, the sunflower star ranged from Baja California down in Mexico all the way up to the Aleutian Islands in Alaska. And they tended to prefer shallower depths down to about 150 feet depth, but they've also been found at depths exceeding 1,000 feet. Historically, they were pretty common across this entire range and were a regular occurrence on dives across much of the region. So now let's talk about the sea star kelp forest connection. As I mentioned, as voracious predators, sea stars can have a big impact on a food web and its associated ecosystem. Since the sunflower star can prey on urchins, they can theoretically promote healthy kelp forests by keeping populations of these kelp munching urchins low. And there have been several studies that have borne this theory out. Several studies have found that when the sunflower sea star was removed from a kelp forest, there were more urchins. Those urchins were out and about on the reef grazing, and consequently there was less kelp. So we know they have the potential to impact the state and resilience of kelp forests, but we don't know how often they actually do this. There are actually a lot of things that we don't understand about the role that sunflower sea stars can play in kelp forests. We don't understand that if there are other predators like otters in the system, do sunflower stars still influence urchin densities as much? If the urchins are starved, as they often are in barrens, will the sunflower stars still eat them? If these stars have access to lots of other prey like mussels and clams, are they still interested in eating urchins? So overall, there's a lot more research both in the lab and in the field that needs to happen to better describe and predict when and where the sunflower star protects kelp forests. With that being said, one thing I want to point out over here on the right side of this PowerPoint slide is that in many places, sunflower stars are one of the only major urchin predators. In North America, we've consistently found that urchin-eating predators like otters and spiny lobster are often important to keeping kelp forests healthy. If you look at this map over here, I've laid out the ranges of the West Coast's major urchin predators. We've got the sunflower stars range in purple, we've got otters range in brown, sheep's head down here in blue, and then spiny lobsters in red. Now, if you're unfamiliar with any of these species, that's fine. Just know that they're all predators that can eat or scare away urchins, and they've been found to promote kelp forest health through their effect on urchins. Now, kelp forests are present along the entire West Coast, going from Mexico all the way up into the Arctic. So if you look at this map, you'll notice that there are large swaths of the range of kelp forests on the West Coast where sunflower stars are actually the only major urchin predator left, or they're one of maybe only two. So in places where you don't have otters or sheep's head or lobsters, historically the sunflower star was one of the only predators left that can eat urchins in high enough quantities to reduce their densities. So there are many places along the West Coast that with the disappearance of the sunflower star, there essentially aren't any major urchin predators left. And as you'll notice from this map, Oregon is one of those places. So those were all the really cool things about the sunflower star. See, there it is, rolling right off my tongue, Pycnopodia. Now let's get to the not so cool part. Many of you at this point may have already heard of sea star wasting syndrome. And this was a massive epidemic that broke out around 2013 and started killing sea stars. Over the course of the next couple years, wasting syndrome struck essentially the entire West Coast, from Mexico all the way up to Alaska, affecting many sea star species and dramatically reducing their populations. Up here, you can see a picture of a wasting sea star, and you can kind of see these lesions on its body and how it looks like it's almost decomposing, which is how we identify whether they're wasting. And to give you an idea of just how quickly this epidemic impacted stars in some locations, these pictures along the bottom here are a picture of a rock in the Salish Sea, shown about 3 weeks apart. Now, on the left, before the outbreak hit, this rock was completely covered in sunflower stars. And then on the right, just a few weeks later, they were just gone. And this didn't just happen in the Salish Sea. This is an observation that many people up and down the coast were having as the epidemic progressed. So it moved with astonishing speed. So just how bad was this crash for the sunflower star? Well, my team, a team funded by the Nature Conservancy, undertook an IUCN Red List assessment. IUCN stands for the International Union for the Conservation of Nature, and they host the Red List, which is an international designation of whether a species is endangered or not. We collected data on when and where the sunflower star has been found from 33 data contributors, which altogether gave us about 61,000 surveys of the sunflower star all across this range, going back to the 60s, actually. We then used this data to assess just how much their populations had been reduced across their range. And we found that across the range, from Baja to Alaska, the population size of sunflower stars has declined by about 91% due to this epidemic. And we estimate that in total, about 5.7 billion sea stars died. And yes, that's billion with a B. These frightening declines were bad enough that they pushed the sunflower star into the critically endangered category on the IUCN Red List, which is the most severe status a critter can receive other than extinct and extinct in the wild. So how bad was this crash? Really, really, really bad. So I said that their populations declined by about 91%, which means that there are some remaining populations. So where are these remaining populations? Well, I can tell you that the species is nearly extinct in the lower 48 states in the United States. We estimated that their populations from Baja to the outer coast of Washington, so not including the Puget Sound, just the outer coast of Washington, have declined by about 98 to 100%. And in fact, they really do appear to be essentially locally extinct in some areas. For instance, of the thousands of surveys that we looked at, since 2015, not a single sunflower star has been found in Mexico. And since 2018, not a single sunflower star in our surveys had been found in California. And since about 2016, 2017, only a handful had been found in all of Oregon. However, as you move north and you get into the Puget Sound, sorry about that, folks. You get into the Puget Sound and the Salish Sea, you start to find robust or surviving populations again. So up north, when you get into the Puget Sound, British Columbia, and most of Alaska, their populations only declined by about 87 to 95%, much less so than down south. Even up north, though, it's really locally patchy where you find them and where you don't. For instance, in some places like Haida Gwaii off the coast of British Columbia, they've barely seen any sunflower stars since the outbreak. And then conversely, we have reports from divers in some of the fjords in the central coast of British Columbia that I found 20, 30, 40 sunflower stars on a single dive. In fact, this picture over here to the left is a picture of 4 sunflower stars taken in one picture on a dive in a fjord in central British Columbia just this year. So that's exciting news. That's good to hear, right? There's still populations in the northern half of their range that appear to be pretty stable, albeit at very reduced densities. So in many places, it's been 5 to 8 years since the outbreak really began. Another question we asked, therefore, was have these populations began to recover at all? Well, based on the 61,000 surveys we collected, we would say no. We don't see any evidence for recovery, even in those places that still have surviving populations. This graph over here on the left that I pulled up is a little difficult to see, but the important part is that along the x-axis, we have time going from 2017 to 2020. And along the y-axis, we have density of sunflower stars in different places. The various lines you see with different colors represent the trends in population size over the past few years in different regions. So each color is a different region. And I know these are a bit difficult to see, but really the only thing you need to notice is that none of these lines are going up. None of them are increasing. So on the outer coast of the contiguous US, California, Oregon, US sunflower star populations have flatlined around 0. And even in the more northern areas where you still have surviving populations, their populations have either remained steady, not increasing or decreasing, or they've been slowly decreasing over the past several years. And the fact that we don't see any kind of recovery yet is a little concerning because other sea star species that were impacted by wasting have begun recovering their populations in some areas. For example, here in Oregon, the charismatic intertidal ochre sea star was hammered by wasting syndrome. But a month ago, I went out to Seal Rock at low tide and I saw plenty of them. They're the big orange and purple sea stars. I saw plenty of them crawling around. So while some other sea star species are recovering, the sunflower star isn't, at least not right now. And so for now, we don't know whether the sunflower star is coming back in the lower 48 states anytime soon. So just to kind of summarize their current status, we saw catastrophic losses globally. They're nearly extinct on the outer coast of the contiguous US. There are surviving populations throughout the Puget Sound, British Columbia, and Alaska. And thus far, we're not seeing any evidence that their populations are bouncing back from this epidemic. So, heavy stuff. It's not fun to hear and talk about. But let's focus to thinking a little bit more towards the future. What can we do now? What are our conservation options? Well, unfortunately, I have to point out that before we can do any meaningful conservation, we need to know more about this species and particularly about this disease. So it's been about 7 or 8 years since the epidemic began, and we still don't even know what actually causes the disease. I mean, it still boggles my mind. Think about that. This was one of the worst die-offs of marine animals in recorded history. Billions of animals died in a few years, and now, almost a decade later, we don't even know what causes the disease. We have to be able to identify the pathogen, whether it's a virus, a bacteria, amoeba, whatever it is that's causing this disease. We have to identify that before we can do any more serious conservation work. Why do we need that information? Well, consider if we tried to do conservation efforts and we spent a bunch of money and energy and people's time trying to reintroduce a bunch of sunflower stars back into Oregon or California, and then they all died from the disease within the first 6 months. That's not a feasible conservation option. And our options are really limited until we better understand the disease itself. So, understanding that research is really the first step to conserving this species, what are some of our other options for conservation beyond research? Well, some of the options that our wonderful team came up with are: one, translocations, which would involve taking stars from surviving populations in the northern half of the range and reintroducing them down south. Two, we could also pursue captive breeding, conversely, which would require finding surviving undiseased sea stars, preferably in the places where you're hoping to reintroduce them, bringing those sea stars into the lab, breeding them in captivity, and then reintroducing those critters into the places missing the sunflower star. This is kind of similar to what they've been doing with California condors, breeding them in captivity and then re-releasing them. In addition to methods of trying to repopulate them, additionally, it's probably very important to try to protect the remaining stars that are left up north. For instance, sunflower stars often get caught in crab traps or on long line fishing lines. And we don't know whether that bycatch is hurting the remaining populations, these really fragile, decimated populations in the northern half of their range. Also, if there are key habitats for sunflower stars, which we still are unclear about, but if they had key habitats like seagrass beds or kelp forests or the fjords up in British Columbia that play an important role for this species, protecting those habitats could be an important way of protecting the surviving remaining sunflower star populations. So those are some of the options on the table right now. I'm sure there are other ones, but those are the ones our team came up with. And I can report to you that the Nature Conservancy is currently supporting captive breeding efforts with the hope of having established lab-bred captive animals should there eventually be interest in reintroducing them. On the kind of individual side of things, one thing that you could do to help us gather information about these surviving populations is that if you ever see a sunflower star out in the wild, you can report it. Try to take a picture of it. Those really help. And report it either at seastarwasting.org or on the website iNaturalist, which is a popular naturalist website, I guess the best way to describe it. So those are some of our options looking forward. Now I'd like to reflect a little bit about what this means for us here in Oregon, where I live and I'm focused on. So I think a few things that the case of the sunflower star bring to mind are one, we're unlikely to see the sunflower star back in Oregon anytime soon without some kind of human intervention. Two, at this point, we've now lost our two most important urchin predators, which are otters and sea stars, particularly the sunflower star. We are seeing increased urchin densities in many parts of the state. Like this picture on the right here, you can see that I took of an urchin barren down in Cape Arago. Four, we are beginning to lose some of our kelp forests to these urchins, particularly in the southern half of the state in places like Port Orford and Coos Bay. And finally, if Oregonians wanted to do something about protecting kelp forest ecosystems, one option would be to try to reintroduce a predator like otters or sunflower stars. Currently, there aren't any groups considering sea star reintroduction in Oregon. However, if you're interested in learning more about otters, I would suggest checking out the Alaka Alliance. It's pretty easy to just Google them online. And they are currently leading the charge as to bringing up the idea of potentially considering whether it's worthwhile to try to reintroduce sea otters back into Oregon or not. So, I'm not here to advocate one way or the other. Predator reintroduction is something that communities need to make decisions about because they have a lot of impacts for other parts of the ecosystem. But in other places, we have seen oftentimes when you reintroduce predators, it can be an important means of protecting and keeping your kelp forests healthy. So that would be one option moving forward if Oregonians came together and decided they wanted to do something about this. So, that was all focused on the sunflower star. And I kind of want to step back for a second and go back to that initial question about what do you think of when you think about conservation? So what does the case of the slimy, crawly, stomach extruding sunflower star show us? I think the first thing it shows us is that, yes, we all love big, beautiful, intelligent mammals, but they cannot be our definition of conservation anymore. When we lose creepy, crawly invertebrates or plants or algae, that reverberates all the way through our tightly interconnected ecosystems, and it affects everyone. We have to move beyond the idea of conservation being about charismatic metafauna to thinking about how do we conserve ecosystems and all the key parts of the ecosystems, even if those key parts aren't necessarily charismatic. Point two, I think that we should start expecting to see more of this, expecting to see more of these sudden, fast-moving, and often catastrophic events like sea star wasting syndrome. And one of the reasons I say that is, think about this. We've seen sea star wasting syndrome before. It's popped up multiple times in California, going back to the mid-1900s, where we've seen sea stars get these characteristic lesions and kind of start decomposing. So this is not a new disease. This is not a COVID that suddenly popped up and ran amok. But something changed, something supercharged this disease that's been around for at least 100 years. And in 2013, during one of the worst marine heat waves in the West Coast's history, this disease went from being a small problem in California to coming close to wiping out an entire species from Mexico to Alaska. Now, we, as in the scientific community, don't know if or how exactly this epidemic was related to climate change. But the scientific community has been telling us for years, as the climate changes, it begins to kick off cascading chains of events, and you start to see surprises. You start to see things change really quickly and suddenly because of these cascading chains, because of these positive feedback loops. And very quickly, you begin to see breakdowns or collapses. And so I just kind of want to stress the point that I want folks to be aware that we're going to start seeing more events like this that challenge the integrity of entire ecosystems. And we need, as a society, as individuals, we need to start thinking about what do we do with that information? We need to brace ourselves and look to the future. And then finally, I think one of the things that for me as a young scientist, one of the things that the case of the sunflower star has really challenged me on is questions about how do we fund science and conservation in the face of a rapidly changing planet? As I mentioned, it's been 7 or 8 years since this epidemic broke out. This epidemic thought to be one of the biggest wild marine die-offs in recorded history. This epidemic where billions of animals died in the course of a few years across an entire continent. It's been 7 years since this happened, and we still don't even know what caused this disease. And conservation efforts are just now starting due to the resolve of a few, of a few determined folks at places like the Nature Conservancy and individuals, who care about the species. This kind of slow, awkward, reactionary response to ecological disasters, it's just not cutting it. Now, I'm not sure how we change the structure of US research and funding to address this. I'm still trying to figure that out myself as a scientist. What can I do? But I know that this isn't working. I know that we need the help of lots of different kinds of people to figure out how to do better. We need fishermen, coastal residents, artists, authors, scientists, politicians, and so many others to start thinking creatively and thinking fast about how do we navigate our changing relationship with nature, and how do we need to change our funding and our science to keep up with that. We have to do this. We have to think creatively and think of new ways. So that being said, I want to go ahead and wrap up here, folks. I know it was a little bit on the short end of the spectrum, but I figure, nobody needs to hear me drone on for that much longer, and I want to make time for any questions. So I just want to really appreciate each and every one of you for making time to come and share your attention and your concern with me. It means a lot to me to see so many people show up, even with technical difficulties. I'd also like to thank the so many folks who were involved in making this work happen. This work is not about me. It was co-led by particularly these three folks up here: Dr. Sara Gravem at OSU, Fiona Saccomano at the Nature Conservancy, and Dr. Walter Hetty, also at the Nature Conservancy. And thanks to the Nature Conservancy for funding this work and Pycnopodia conservation in general. I'd also like to just point out that, I mentioned those 33 data contributors. Well, those people are all listed over here on the right, and others. So many people came together to share their knowledge, their data, their insights with us to try to make this red list assessment happen. And it was amazing. I feel so grateful to have been a part of that. And I really want to acknowledge that these people made this stuff, they made this possible. Down below, if you have questions, if you have data, if you're interested in learning more about the recovery team, my email is hamilton@oregonstate.edu, and my Twitter is @SarahHamilton593. And I'm always excited to hear from people. So with that being said, we can open it up to questions now, Cinnamon.
That sounds great. I want to take just a second. That was amazing. Thank you for sharing this. I know this is a hard story to tell, but I was really interested in learning more. Before we jump into questions, because they're coming in quick and fast, I just want to let folks know that joined us a little bit late because we had trouble with the link. Thank you so much for finding us and finding the way. If you didn't hear the beginning, the recording of this seminar will be posted on the HMSC past seminar page in a few days, and you can watch it there. You can also share it out with others if you find others that weren't able to join us, and I just put that link where it's going to be back into the website. So Sarah, I'm going to work through some of these questions, see if I can start at the beginning. I think the first one that I saw come in was, how do you envision a captive breeding program if we don't even understand how the pathogen works? So can you talk a little bit more about that?
That is an excellent question. I think one that we are currently grappling with. Now, one thing I'll point out is I am not an epidemiologist, so I don't have a deep understanding of the way diseases work. But I do know that one of the reasons we haven't identified it yet is that it happened so quickly and across the entire range so fast that by the time people started trying to do research on it, there was no way to tell whether the sea star that you got had already been exposed to the pathogen or not. So you would bring supposedly clean, supposedly unexposed sea stars, sunflower stars, into the lab to try to test. And before you could even introduce a virus or anything, they would start wasting. Where did that come from? Sea stars in aquarium that had never been, that weren't out in the ocean started wasting, probably related to the movement of, water exchange between aquaria water and the ocean. So the question is, I know that these captive breeding efforts are working. Essentially, we've got, I think the biggest juvenile sea stars right now are like about that big. We've been working on this about a year. So it is working. We're getting some juveniles to breed and start growing in the lab, and we don't know if these are sea stars that were never exposed to the disease, so that's why they survived and they're doing well, or if conversely maybe they were exposed to it and there's some gene that confers resistance to the disease. Um, so I know it's working, even we are able to establish some kind of captive breeding without knowing what the pathogen is. But the next step of then, you know, we don't know that these stars that we've raised in captivity, we don't know whether they have the disease or not. And so currently taking these stars and trying to reintroduce them into Oregon, we could be reintroducing a pathogen into the system. And so that is, I'm sorry, I don't have a better answer. It's a huge mystery and another reason why we can't move forward with good conservation until we understand just the basics of this disease.
So there's a lot of questions, but I'm going to jump to one that's kind of related. Can you talk a little bit more about the larval stage, how long it takes for these sea stars to become reproductive, how they spread their larvae, and what are settlement cues, trying to understand if we have a dispersed population, if they're settlement density dependent with their reproduction?
Yeah. Those are great questions. Short answer, we have no idea. Um, Unfortunately, because this species is a creepy crawly invertebrate, and because historically it wasn't a harvested species, a lot of people haven't. And one other thing, they're actually surprisingly hard to raise in the lab. They don't like being in aquarium and being in labs. They tend to die. And so because of these reasons, it's hard and they're less appreciated species. Historically, we haven't done the research. And so I was in charge of trying to assess generation time for the IUCN analysis. Just a basic question of how long do these critters live? I spent probably a month searching every bit of literature I could find. And the answer is we really have no idea. It's probably a long time. But the same kind of answer goes for this question about the larval stage. Gosh, I I'm not able to remember what how long the larval stage lasts for most sea stars. I'm guessing it's somewhere in like the 20 to 50 day range, but we have no idea really what it is for Pycnopodia. We have no idea what their dispersal patterns look like. We have no idea what are their cues to settle. So again, how do you go about conserving a species that you don't even know how long it lives? You don't even know what kind of habitats it prefers. So I hope not too many of these questions I have to say we have no idea, but that is part of the reality and is very frustrating from a scientific point of view and it's also a little embarrassing, like I don't like coming here as the expert on sunflower stars and having to admit that I don't know any of these things. So it's a frustrating situation.
So you talked a little bit about that it wasn't a harvested species. I have a question here that said that they were shocked that ODFW still allows 10 sunflower sea stars to be harvested. Do you know about the harvesting of allowment for ODFW in Oregon?
I don't know about that specifically. My guess is that I don't know of anybody who harvests pycnos, sunflower stars in Oregon. And so my guess is that it's an old law on the books that ODF and W established back before there was a problem. And they said, well, just don't take too many. And now that things have changed, there's such little interest in harvesting them that ODF and W hasn't taken the time to update that. We're considering, one option is to potentially try to list them as an endangered species under the Endangered Species Act in the US, which would create penalties for harvesting them for any reason. But it gets really tricky because they crawl into crab pots, right? When you go out trapping for Dungeness crabs, Pycnopodia will oftentimes crawl into those pots and get pulled up. And then if they're listed as an endangered species, are the fishermen that accidentally pull one up, A, we don't know how much it hurts the star, and B, are the fishermen then going to face some kind of repercussion because they may have harmed an endangered species? Um, so, but specifically on the ODF and W front, my guess is that there's been so little interest in it that they haven't found a need to change the regulation because they just don't see it happening.
Thank you for that. I have a question here that's a little bit interesting that talks about, couldn't it be a little bit more similar to COVID than we imagine? COVID's been around in the past as well. We just have a different variant. It could be that this is a different variant or that it's possibly the same one with just a new mechanism of spread.
Yeah, that's a great point. You're right. And I probably put my foot in my mouth regarding COVID. My understanding was that, there are similarly related coronaviruses like SARS and MERS, and that something changed to create COVID. But you're totally right. It could have been, for instance, there's a new variant of COVID that has been found in the UK and other places that has increased transmissibility. It's much easier to pass from one individual to the next. And that could have been something, that could have gone on here as well. So you're very right. We don't know what this had to do with temperature or with climate change. We don't know if the virus itself changed or if the environmental conditions changed and made it more aggressive or something like that. So yeah, sorry, I don't have a good answer on that one.
So here's another one you might not know the answer to. I'm just going to hit you with them, Sarah. I'm sorry. Do you know about the genetic diversity within Pycnopodia? Are there unique subspecies or anything like that? And so is there like a potential that there's a deep water population that's maybe different than what we're seeing? Or
Yeah, excellent question. We've been asking ourselves that as well. I will point out that on this list of contributors, you see right here, Lauren Schiebelhut, right in the middle there. She's a researcher at UC Merced who's investigating Pycnopodia genetic diversity currently. But again, we don't know. And one of the problems is now their genetic diversity has been so completely altered, decimated, changed by wasting that we can't study what their genetic diversity used to look like, how much genetic diversity there used to be, what the subpopulations were, and compare it to what it is now. She's kind of just has to work with what specimens she can get now. So yeah, that one's also a black hole.
Can you speak a little bit to, we all heard about the adenovirus kind of at the beginning of all of this. Can you talk about how that hypothesis has changed or where we're at with understanding what the pathogen is?
Oh, Lord. I will certainly attempt to. It is a fraught field right now. As a non-epidemiologist, I just kind of step back and watch it all happen. But yeah, so our initial hypothesis was that it was a densovirus, which was a virus that was often found, I believe, associated with the lesion tissue. So these lesions that they developed as they were wasting. I think a couple things that threw kind of a monkey's wrench into that hypothesis were that we found that the densoviruses that we thought might be causing it are also present in non-wasting populations. So it could still be that it's a densovirus. It's just that something has to trigger them or change. But when you see that virus in non-wasting populations and in wasting populations, that makes it harder to say that that virus is causing the disease. So, and another thing that's really difficult apparently about lab experiments on wasting is that so many times you bring ostensibly healthy stars into the lab and they waste. They waste if you increase the temperature. They waste if you decrease the oxygen. They waste if you add a densovirus. They waste if you add a different kind of virus. So it seems like there's something going on with when they are stressed, they are more likely to be wasted or to waste. And when you bring them into the lab, they're automatically stressed. And so there's actually a really recent paper just came out that y'all might want to follow. It was published in Frontiers in Marine Science. And it was an interesting theory that basically like proliferation of bacteria on the skin of these Pycnopodia caused low oxygen. Anaerobic bacteria proliferate. They cause localized low oxygen, and then that causes the lesions. But I can tell you that that's a very controversial theory, and I think you'll be seeing some controversy coming out about it because I know someone who's just about to publish something critiquing that paper. So, um, yeah, right now we've got that one hypothesis about maybe it's low oxygen combined with bacterial proliferation, and then we've got the densovirus hypothesis. Neither one of those are really, you know, neither one of those are really fitting all the bills or really passing all our tests for being, definitively proving what it is. And so I think another thing that's difficult is I think a lot of researchers are hesitant to take on trying to do this project, and a lot of funding agencies are hesitant to fund it because people have been trying and it's really hard to do. So it's harder to find funding to go do this necessary research.
Do you have any information about current breeding programs that are trying? Contact information if somebody wanted to learn more about how the procedures that people are trying?
Yeah, that's a great question. I so the Nature Conservancy is supporting that effort. I currently only know of one captive breeding effort. It's based out of the University of Washington. I don't know of any others. If you wanted to learn more about it, I would say either email me and I can give you the specific email addresses, or I would say email the Nature Conservancy with a general inquiry. And you can mention Vienna Sacamano and Walter Hetty, who work at the Nature Conservancy and are involved in this project. They would be able to talk more about all the hard and all the good work that TNC is doing to support captive breeding, as well as some other reintroduction or some other conservation options. Mm-hmm.
And just a reminder, Sarah's email is on the screen now if you wanted to reach out to her directly. We're still getting lots of questions, so if you're still in there, we'll do a couple more here. Are there any studies on the immune systems, or can you speak to the immune system of sea stars, either Pycnopodia or others?
Ooh, that is a great question. I feel very positive there is research on immune systems and sea stars. Um, I unfortunately I don't come from a sea star biologist background. I come from like a kelp and urchin background. And so I'm not actually that up to date on their physiology. So that's a great question and something I should look into myself. I know for invertebrates, it's totally different than it is for vertebrates. And I don't think they have the kind of immune system that can like have any kind of immunity, as in like be exposed to a threat and develop some kind of warning system for when that threat is thrown at them again. I don't think they have that kind of adaptive immune response. But beyond that, I don't know that much about what kind of immune system they have. So I'm sorry about that.
I'll take you to a question maybe you can speak to. You talked a little bit about using the citizen science program. There's a question here about, can you speak a little bit about how to better mobilize citizen science to provide more useful data for monitoring recovery? So can you go into that a little bit more?
Yeah, that is an awesome question. First thing to note is that of these 61,000 surveys, probably something like 10 to 15,000 of them came from citizen scientists. So there are particularly organizations like Reef Check and Reef and the Center for Alaskan Coastal Studies. There are some awesome organizations mobilizing citizen divers, particularly, as well as citizen intertidal scientists going out and doing some of these surveys already. So I would encourage you to look into Reef Check, and the Center for Alaskan Coastal Studies. In terms of mobilizing citizen science to monitor recovery, there's so much that could be done for this. Like people really care about this species and they want to go find it and tell us about it. I think honestly, I think two things. Honestly, there would just need to be some funding and some organization. Like if you could pay somebody, for 6 months to organize some kind of centralized system where people can report things. That's kind of what we were trying to do with the iNaturalist project and the seastarwasting.org. It's just a matter of gathering all those observations from all these different people. But the other thing that I think of, and I think would be an amazing route to take is utilizing the fishing community, right? The fishing community's out there all the time, putting out crab traps, catching Pycnopodia in traps, catching Pycnopodia on long lines. And so two ways that we could utilize fishers and get them involved in this conservation effort is, if they ever catch a Pycnopodia as bycatch, if there was some sort of app that you could just easily ping it and, it would send a picture and your lat long to iNaturalist or something like that. And then additionally, I know, I was talking about captive breeding. And if you want to establish a captive breeding program for, say, Central California, you would kind of prefer to start with sea stars from Central California. And so if there are surviving individuals out there in Central California and Oregon, I think it would probably be fishers that would find them, right? Because they're out there way more than scientists are. They're putting 1,000,000 long lines out, 1,000,000 crab traps. So we've talked about the idea of potentially kind of putting out like a bounty for Pycnopodia. If someone really wanted to start a captive breeding program, letting all the fishermen know that we'll throw $100 your way if you can bring us an intact Pycnopodia, something like that. So yeah, I would love to be able to involve the fishing community in some of this work as well. They're such a wonderful resource.
Nice. So we're running close on time, but there's a couple of questions that are coming in about, you talked a lot about ecosystem, but then you kind of talked about introducing an individual species back in. Have you all talked about what a species kind of multi-species introduction might look like? So thinking about how to remove urchins, how to bring in the otters, and how to deal with kelp in kind of a collective? Or is that just too hard to even kind of comprehend because we have these interlocking issues? So I was just wondering if you could speak to that a little bit.
Yeah, I mean, it's a fantastic question. I don't know very many places where multiple species have been reintroduced at similar times. That would just be mind blowing to do. And one thing I will note is that, I've been studying kelp forests in Oregon for about 4 or 5 years now. And it's only really in the last 2 or 3 years that I've seen people really start talking about kelp forests. We are right at the beginning edge of having the community really engage with these questions and talk to each other and talk with scientists, and scientists talk with the community and talk with fishers. We're right at the beginning. There's so much momentum, there's so much infrastructure, there's so much building that needs to be done. So I would say that definitely hasn't been considered. We're still kind of like, it's we're so early on and even thinking about, do we want to try to reintroduce a predator species? That the thought of trying to introduce two is just, we're not there yet. But it would be fascinating scientifically.
So coming at it from the other side, there's a couple of questions here about calling of the purple urchin, how it's done and its effectiveness and how that works and whether it will have an impact. Do you want to speak about that a little bit?
Yeah. So there's a lot of interest in urchin calling. I think it's partially because it feels like something we can do here and now about the problem. And I will say, urchin culling we're seeing some experiments come out now that show that it can be really effective, and it can, reduce the number of urchins, increase the number of young kelp that are able to grab onto the substrate and grow before they get eaten by urchins. So we're seeing that it can work. It takes a lot of time and energy and money. There are millions of urchins out there on Port Orford Reef right now, one reef. And so I don't think, as in terms of human-based culling, I don't think, unless, there were to become like a commercial market largely for purple, big commercial market for purple urchins, I don't think that urchin culling is a solution on a big scale. It may be something that you do for a particular cove, either because you really care about that kelp forest or because you want to establish little seed populations that can, maybe repopulate other areas. But it is, it just takes a lot. It takes a lot to do it and keep those urchin populations low enough for long enough that the kelp start to come back. And I'm noticing some of the comments. If you're interested in this, I would go Google the Oregon Kelp Alliance, or maybe get in touch with Tom Calvanese down at the Port Orford Field Station. So there is a very recently established Oregon Kelp Alliance that is considering trying to get some urchin culling experiments started down in the Port Orford area, and to use the urchins that are culled to be sold off for uni and things like that. So people are considering it, but it takes time and energy and manpower and organization. And right now, nobody's funding any one person to do this. So this is all of our side jobs. And that's just not how a big project like a good urchin removal effort goes.
This has been an amazing conversation that is going on in the chat and with you, Sarah. Thank you so much for coming to share with us. For folks that are still chatting in questions, if you have anything that I definitely jumped around. I did not answer every single question, and I apologize for that. So if you have something that's burning that you weren't able to ask in this format, please, I'm going to throw Sara under here, email Sara directly and ask your questions to her. If you can also just help me out for those folks that missed tonight's seminar, really share out that we're going to post that recording in a few days on the past seminar page. And I apologize so much for the inconvenience with the technology. Sometimes things happen, but thank you so much for finding us, and thank you for joining us. And Sara, thank you so much for sharing your work with us. It is just critical that we start to think about this and we think about things a little bit different. I think you can see the chat, lots of thank you very much. If you could hear it, we're all applauding and thanking and standing, and I'm just really happy you shared your time with us on this rainy, stormy beginning of 2021 day.
Yeah, this is the best part of my job, getting to talk to, to the public and to fishers and things like that. So thanks to everyone for showing up, and thanks to Cinnamon for her last minute efforts to make this happen. I'm so glad it turned out to be worth, we got so many people here despite everything.
Yeah, thank you so much for checking your emails and following new links and just being patient with us. It was a bumpy start, but we had a nice turnout, and so thank you very much. Again, we're still getting lots of thank you. Great sharing, so thanks very much. All right, everybody, I hope we see you in February at our next Science on Tap. Everybody else, again, share that link out when I get it posted. And Sara, thank you so much for your time and your effort, and good luck on your continued work.
Thank you so much, Cinnamon.
All right, everybody, I'm going to end the presentation. Thanks so much.